What is the difference between Eminem and Miley Cyrus? I gave you a clue here. Eminem has balls and Miley Cyrus does not, right? So what exactly does the balls do? So what can Eminem do what Miley Cyrus cannot? The balls, also known as the testes, is the place where sperm is made, right? Sperm is the male sex cell. This is what's gonna pass on Eminem's DNA, Eminem's blonde hair. So we call this process of making sperm spermatogenesis, and I'll cover that process in a separate video. Now, the female version of balls is called the ovary, and what it does is it makes the egg in a process called oogenesis. And again, this process of oogenesis will be in a separate video. Now, it doesn't make this kind of egg, right? It makes an egg that is a cell. Just like sperm is a cell, egg is a cell. Now, bear in mind that sperm is about this small compared to an egg, so don't get that confused. But the point is, Eminem's testes will make this sperm, which carries half of his DNA. Miley Cyrus's ovaries will make this egg or this ovum, which carries half her DNA. And when this sperm and egg meets, let's say they meet, sperm will pass on its DNA to the egg in a process called fertilization. Now again, this process I will cover in a separate video. But when this happens, this egg will now receive the sperm's DNA, or Eminem's DNA, to form the normal amount of DNA, right? Because half DNA plus half of the DNA will make the full DNA. Now this cell will replicate and grow in pregnancy to a full-blown baby, which will resemble both Eminem and Miley Cyrus in some ways, right? Depending on which traits. And again, this part, pregnancy, I will make a separate video for. Now, what am I talking about now? What's the purpose of this video? We're going to focus on the structure of sperm and the structure of the egg, okay? We're going to focus on both of the key structures that you need to know. Not how they're made, just what structures you need to know. So here we have sperm, okay? Obviously, this picture I have is far more complicated than the sperm I have here, right? But let's just look at it. It's quite simple. Notice first how complicated this is. One cell. Can you believe it? It's one cell. Because normally when we think of cells, we think of like a round structure with some obvious nucleus and organelles, right? It looks real simple. But don't get it wrong. Cells can have crazy looking shape, just like a sperm. Now, a sperm can be split up into three categories: head, middle piece, and tail. Head, middle piece, and tail. Some books will tell you there's a neck piece as well, but for the IB, this is good enough. So now, what are some key things? Look at this head. What do you think this is? This is a nucleus. This is where the half of Eminem's DNA is stored, right? That's real simple. What is this part here? This is a little sac or vacuole, a bag that contains a lot of enzymes, and enzymes are known to break things down or to make things, right? They're like little tools. Now in this case, these enzymes here are meant to break things down. So we call this little sac here the acrosome. The acrosome. Don't worry about the functions. I made a table here after I explain each structure, and you can come and check it out and it'll be clear right after. So that's it. So what's the purpose of this? Why do we need enzymes here that can break something down? Remember, when the sperm meets the egg, it's got to penetrate it. It's got to burst right through it so it can pass on its DNA into the egg, right? So that's why we need these enzymes that can break things down. So that's basically it for these two. Now, what about this? What is this one right here? This is a tail, right? And we call it by another name, a filament or a flagella, right? And guess what the purpose is? To swim, right? It's like using an oar on a canoe. It helps you to row. It helps this sperm to move around because it's got to swim towards the egg, remember? That's the basics for that one. What about this? What are these little things? These are mitochondria, right? That's an organelle you can find in almost all cells. Mitochondria. And what does it do? It makes ATP. ATP is like energy for a cell. Why do you think the sperm needs energy? Because it's got to swim, man. It's got to go fast. Without all these mitochondria, this flagella ain't going nowhere, right? So this is very important, mitochondria. The more mitochondria, the better. The faster this sperm can swim, the more likely it will be the first one to win the race. Now, lastly, we have this one, centriole. This one I would say is not so important for you to know, but I'm just going to put it here and list some. It has various functions, but they're not crucial for you to know. If you've heard of a centriole before, you know a centriole is a little thing inside a cell that helps it split apart in mitosis. So when the sperm meets the egg, okay, let's go back here. When the sperm meets the egg, it will pass on this centriole, which will help the egg or the baby cell to divide and divide and divide, right? So that's one of the key functions. And another function is the centriole is like a little thing that connects the head and then the lower half of the sperm. It's like a connector. And then lastly here, it has another one. It also controls beating. So it's like a controller. You know how the tail swims? This thing is a little bit in charge of controlling the tail from moving. Okay, so it's a controller in a sense. And then finally, this one again is a small function. It can, when it's passed on to this cell here, remember, when it's passed on to this cell, it can help maintain the structure. So it can help make this cell strong and stable for the baby to replicate and become bigger over time. So again, it's got a bunch of functions. It's in this table if you want to check it out. And again, not that important. So that's it for the sperm. What's next? Let's go to the egg. Here we got the egg. The egg is pretty much the target site of the sperm. The sperm is going to swim towards this egg to try and fertilize it. Now, let me copy the spermie to make this clear. So the ultimate objective of the sperm is to come, penetrate, release its DNA so that this egg now has the mom's DNA or Miley Cyrus's DNA and also Eminem's DNA. So it got the total DNA to form this normal, healthy baby, right? So what's the first problem that sperm faces? It swims and it swims. Oh, it's got this outer layer. This outer layer is made up of cells. So this is not really the egg. This is something surrounding the egg. The egg is one big cell, this thing right here. But this outer layer is a bunch of cells that kind of protect the egg a little bit. So it's not so easily penetrated by sperm. So we call this layer the corona radiata. Besides being, so it's not part of the egg, it's surrounding the egg. Now, besides protecting the egg a little bit, it also provides nutrients like proteins and things like that to keep this egg healthy. So it's going to send proteins into this cell, keep it healthy, right? That's basically it. Next, we have this slightly inner layer called the zona pellucida. Now, this is starting to be right there at the membrane of the egg. Now, the zona pellucida also has crazy interesting function. So you know how technically sperm from any species can enter any organism, right? So normally a male human and a female human will reproduce, right? Pass on the sperm will come, enter the egg. What if, for some bizarre reason, sperm from another organism entered Miley Cyrus? What's useful is this layer, this zona pellucida, is going to prevent that sperm from another species to fertilize this egg. This layer is very good at distinguishing if sperm is from the same species or not. If it's not from the same species, it simply won't allow it to penetrate. So that's one of the key functions of zona pellucida. And we call that species-specific fertilization, meaning it only allows the same species to fertilize it. Another function is when the sperm from this human organism fertilizes or comes through this corona radiata and then bites onto the zona pellucida and penetrates it, as soon as this happens, the zona pellucida will solidify, become rock solid. The purpose of this is to prevent any other sperms from getting in. As well. Because ultimately, we only want half the DNA. We don't want DNA from so many sperms. We only want one sperm to succeed. So that's why it's so useful. As soon as the first one passes through, it will rock solid. Now, if any other sperm tries to get in, it can't. It's rock solid now. So that's another function of the zona pellucida. We call this preventing, let me get here. Where is it? Oh, no, wrong place. We call this preventing polyspermy. Polyspermy means getting fertilized by many sperms. So this zona pellucida will prevent that. Okay, now we get to some slightly easier ones. Nucleus, right? Nucleus, that is where the DNA will be stored of Miley Cyrus. And again, it's half the amount of DNA of a normal cell because, right, we want half and half to combine, forming a full. So we call this haploid DNA. Then we have this part right here, which is the cytoplasm, right? Cytoplasm is pretty much just like a jelly substance that stores and holds the nucleus and all other things inside the cell. It's just like a little bath, okay? Now, the last one, which will confuse you the most, but don't worry, this one will make the most sense when I explain it in its own video in oogenesis, because in oogenesis I explain how the egg is made. And once you watch that video, you will see where this comes from and how it's formed. But for now, I will give you just a small, quick idea of what it is. Basically, before the egg is mature, so this is an immature egg, it has double the amount of DNA compared to a normal cell. So our normal cells have a certain amount of DNA. Now, this cell will have double that. So once it goes into meiosis 1, because now we're trying to form a normal egg. So it will go into meiosis 1, okay, which is, don't worry if you don't get this, again, I'll make a video on that. It will split into what we call it's slightly more mature now. So now we have, you see the DNA is half, the nucleus has half the amount now. This is now the equivalent to the amount of DNA in a normal cell. But the ultimate goal, right, is to make half the amount of DNA of a normal cell. Now, where did the other half of DNA go? Not into a new cell. The thing didn't split into two cells. The other half of DNA went into a little compartment called a polar body. So we call this a polar body. This is where the other half of the DNA goes when the cell splits. Because the cell doesn't, the egg cell doesn't really split into two. The nucleus splits into two. Because normally in meiosis, the whole cell splits, right? Like for sperm, that's how it happens. Now, next, we go into meiosis 2. So now we have one polar body. Meiosis 2 will be another splitting. So this nucleus will now split again, creating another polar body. That will give us 2 polar bodies total. But then this polar body will also split into half. That will give us what? 3 polar bodies. So overall, we will have 3 polar bodies. And then the nucleus will be haploid, which is what we wanted. And that means when M&M's DNA comes, so the sperm comes, M&M's DNA or sperm will donate his half DNA. And when his half and her half DNA come together, that is a normal amount of DNA. This will develop into a baby. So pretty much a polar body is just the remnants of the DNA that was split. It's where the DNA, it's the extra DNA that we don't need. And these polar bodies will end up disintegrating and going away, and we will only be left with the DNA from M&M and the mom, right? Forming the full amount or diploid amount of DNA. So again, here's a table that summarizes all the key functions that you need to know. I hope this video was useful, and I really recommend, check out these videos that I will make soon.